Hi everyone, my name is Aliyah Rasheed and this is my summer research project, investigating mitotic checkpoint function by genetics epistasis in fission yeast. Let's start by understanding what transcriptional regulatory networks are. These systems are a network of transcriptional interactions that control which genes are turned on or off in a cell. At the core of these networks are transcription factors, which are proteins that control the rate at which genes are transcribed. They determine which genes are transcribed and expressed and play a key role in gene regulation. In this project, I used pombe or fission yeast. It is a simple eukaryote organism that is an ideal model for studying cell cycle regulation. It has approximately 5,000 genes that are regulated by 100 sequence-specific transcription factors. Pombe divides mostly symmetrically, with a cell cycle that is comparable to higher eukaryotes, which means that it shares many similarities with human cells. My focus was on the G2M checkpoint, which ensures that DNA is fully copied before mitosis. One major transcription factor in pombe is called TOE. Overexpressing TOE results in cell cycle defects, and TOE7 in particular is the focus of this study. When you overexpress TOE7, it causes cells to divide too early, producing mononucleated septated cells, resulting in cells that are cut in the middle but have only one nucleus. This is called the cut phenotype. In my study, I looked at 5 putative target genes of TOE7 as seen here. I had 2 aims. First, measure how often the cut phenotype occurs when TOE7 is overexpressed, and second, test whether deleting these target genes could suppress the defective phenotype. In aim 1, TOE7 was overexpressed in yeast cells and grown on EMM plus AU plus T plates for about 24 hours. The colonies were then picked and inoculated in liquid media that lacks thymine to turn on an essential promoter known as the NMT, or no message thymine promoter, which turns on the TOE7 gene when thymine is absent. The cells were grown for 20 hours to reach the exponential phase of the cycle and were then fixed in views using fluorescent microscopy to observe for cell cycle defects. The results showed that the cells with the empty factor, the control, had 0% cut phenotype, but when TOE7 was overexpressed, 54% of the cells showed the cut phenotype. This shows that TOE7 overexpression results in a significant amount of cut phenotype compared to the control, and that TOE7 overexpression clearly causes this defect. In aim 2, we ask what happens if we delete a target gene that TOE7 normally activates. Normally, when TOE7 is expressed, it activates the transcription of a target gene, leading to protein production. This results in a normal or wild-type phenotype as seen here. However, if TOE7 is overexpressed, too much of this process happens, leading to the mutant or cut phenotype. However, if we delete the target gene, we interrupt this process. No transcription, no protein, and potentially no phenotype. And that is what we saw. In all 5 target gene deletions, the frequency of the cut phenotype dropped significantly, even when TOE7 was overexpressed. This suggests that the target genes play an important role in causing the phenotype, and that removing them can suppress it. In conclusion, TOE7 is a transcription factor that delays this final step of cell division until the right time. Overexpressing it causes cells to divide incorrectly, but by identifying and deleting TOE7 target genes, we can suppress this defect and better understand how the cell cycle works. Since similar checkpoints exist in human cells, studying these pathways in yeast helps us learn more about how cell division errors might contribute to diseases like cancer. In the future, we plan to overexpress the target genes themselves to see if that alone causes the cut phenotype, which would further confirm the role in this pathway. I would like to thank my PI, Dr. Chua, for the opportunity to work in his lab, and my graduate supervisor, Leo Zhao, for his invaluable guidance. A big thank you goes out to NSEC for funding this research, and to my lab mates and fellow students for their support along the way. Thank you so much for your time, and thank you for listening.